welcome to springtime officially, and to midterm, the week of midterm vacation. As you can tell, there's no hieroglyphics to be erased from the board, so the students are all gone this week. I'm Dr. Mark Brannon. I'm the president of the board of the Princeton Adult School, and I welcome all of you here. It's great to see you turn out on a beautiful spring evening. It's really a pleasure for me tonight to introduce Professor Manuel Yenes. That's how you spell his name, so I apologize if I didn't pronounce it correctly. Professor Yenes is the Associate Professor of Molecular Biology and the Louis Sigler Institute for Integrative Genomics. He received his PhD in 1999 in molecular and cell biology at University of California at Berkeley. He did postdoctoral work in the lab of Joseph DeRisi at the University of California, San Francisco, and joined the Princeton faculty in 2005. He has a multitude of interests, but we are fortunate tonight that one of his major interests is in malaria. In 2006, he received the Merck Irving Siegel Memorial Award from the American Society of Microbiology, and has received numerous other awards and has published many papers and articles. His research interests include malaria, as I mentioned, parasitology, the regulation of parasite development, host and parasite metabolism, and here's a word that was new to me, metabolomics. And I had to actually go to what everybody in middle school and onward does, and I use Wikipedia to get the correct the definition of metabolomics. It's a scientific study of chemical processes involving metabolites. Well, that doesn't say much, but then it goes on. Specifically, metabolomics is the systematic study of the unique chemical fingerprints that specific cellular processes leave behind. The study of their small molecule metabolite profiles. So that's one of his interests. Molecular mechanisms of malaria parasite, Plasmodium falciparum, is really the area that got us interested in asking him to be one of our speakers. As you know, coming up, we have a number of other great speakers in this series. In 2 weeks' time, bring especially to your attention that President Shirley Tillman of the university will be speaking here. The title of tonight's talk is Combating Malaria, New Ideas for an Ancient Problem. Please join me in welcoming Professor Manuel Yenes.
Okay, is this on? You can hear me? Excellent. First of all, thank you very much, Mark, for the kind invitation, and thank you all for coming today. And thank you for allowing me to lecture on my 1 week off in the spring. Okay. So that being said, what I want to cover today is to really give you, this is not going to work, okay, a lot of background on malaria, and I hope to sort of just generate a discussion. If you have questions, please stop me. I'm happy to answer anything that will come up along this talk. And then ultimately, I want to give you a sense, once you have this background, of what it is my lab is actually doing. And that's the real challenge. As Mark pointed out, I need to convey to you what metabolomics actually is and why I might actually care to use something like that to think about this problem, the malaria problem. Okay, so who knows what malaria means? Where does this word come from? Yes?
Bad air.
Bad air. Okay, so that's the original derivation of this term. Because it was associated with the miasma in Italy. This is an Italian word. We'll do a little bit of history here. This disease has been around for a really long time. All these people that you see up here, historical figures, American figures, people from all over the world, have suffered for eons from this disease. Okay, this is one of the most ancient diseases that we actually know about with regard to its interaction with humanity. And in fact, what troubles me and puzzles me about this is why we haven't come up with solutions that we can actually broadly apply worldwide. Now, I'm going to give you a little analogy. How many people fit in a 747, more or less? About 350, exactly. Okay, so now let's think about the following. Let's think about waking up every day, turning on your radio, and hearing that 7 of these 747s filled with young children has just smashed into the side of a mountain. Okay, this is actually occurring every day with malaria. Okay, this is the current death rate for malaria worldwide, with an annual toll of about 1.5 million individuals. And it's specifically young children, and mostly young children and women. Okay, women during pregnancy. So pregnancy malaria and young children. So the burden of malaria is much greater than this. It's somewhere around 500 million people. 500 million people are getting this disease every year, leading to these 1.5 million deaths. Translates into a child dying about every 30 seconds. And in fact, there's a lot of complexity associated with this disease because it's not just this parasite that I'm going to tell you about, but it has a very complex life cycle involving mosquitoes. And nobody likes mosquitoes. And this is a particularly bad instance where mosquitoes affect humanity's well-being. In countries where this is a problem, it obviously has a large economic burden, and so there's a lot of interesting aspects of disease and well-being for particular nations that are in these zones that have malaria. We are clearly not one of these nations. This is primarily associated with the third world. And the main problem facing malaria today is that it has widespread drug resistance. There have actually been fairly good drugs in the past against this disease, and I'm going to try to convey to you what some of those are and why we've ended up in the situation where we are today. And part of it is that there never have been very many drugs available. Okay, that's a large part of what my efforts are targeted toward. There's no effective vaccine, and I'll keep saying this over and over again. The New York Times loves to report about these vaccines that are coming out. There is no vaccine, bottom line. Okay, nobody you know has ever gotten a vaccine, and we're pretty far away from it. But I have some interesting things to tell you about vaccine ideas. And lastly, in addition to having very few drugs, we don't understand a lot about this parasite, which means that at the end of the day, we have very few established drug targets. So we don't know how to actually undermine the parasite. And I think the big reason is that we don't understand a lot of fundamental aspects of its life cycle. How does it grow? And what does it do to divide? And if we understood those things, we could maybe design better drugs. Now, it's a third world disease, but we're not actually immune to this as Americans, okay? Recently, in 2003, there was a massive outbreak with American soldiers in Liberia, mostly because they failed to properly diagnose a situation that was a clinical situation that was occurring. And secondarily, due to a compliance issue in that the anti-malarials that exist are pretty nasty. They have bad psychoactive side effects, and they can have no effect on you, or they can have really severe effects. And you never get tested before you get deployed. You're just told that every day you're going to take these things, and the soldiers are afraid to take these pills. Okay, so they don't take them. We got all these soldiers that came back and came down with malaria. Okay, a little bit about the disease. So the disease, when you clinically present, in fact, it's very much like having the flu. You have fever, you have headaches, nausea, vomiting. So in fact, for quite a number of days, you don't have a good sense that you have something other than the flu. And then all of a sudden you start having these periodic fevers. Okay, these periodic fevers with the most severe type of malaria will happen every 2 days. Every 2 days you have a fever, and I'll describe why that's the case. This fever ultimately is followed by these periods of shivering and perspiration. So you go hot, cold, hot, cold, hot, cold, in this cyclical pattern. And ultimately, if not treated, the main problems that are associated with this clinically are that you end up with cerebral malaria, and that's because parasites eventually get into your brain and cut off the circulation, and secondarily, anemia. These guys are living in your blood, and they're completely demolishing your blood supply, and I'll show you how that is happening. With regard to severe malaria, so this is sort of late stage aspects. What is actually going on? This is almost exclusively associated with a particular type of parasite that I'll tell you a little bit more about. The fatal symptoms really include, typical things that are associated with severe anemia: liver failure, renal failure, severe anemia, low blood sugar, lots of just sort of metabolic processes that are going awry. Okay, so we've been working on this disease as a community of scientists for easily over 100 years. Okay, how many Nobel Prizes have been awarded either for direct work on malaria or something associated? Anyone have a guess? 0. Well, there's a lot of blank space. Anybody else? 4. Okay, I like 4. Okay, let's see what these have been awarded for. First, in 1902, Ronald Ross demonstrated the malaria parasite was actually transmitted from infected patients to mosquitoes. So this is a seminal observation. Okay, it nullifies the idea that this is somehow coming out of the water or out of the air. Excellent observation. We next learned in 1907 that parasites are actually in the blood of malaria patients. So we first knew about it in mosquitoes before we actually knew where it was hiding out in the humans, the patients themselves. Part of that is because actually a lot of the early research was done in avian malaria. So birds also get malaria. So a lot of the early work was done in birds. In 1927, if believe it or not, the Nobel Prize was given to Julius Wagner Jauregg for the treatment of dementia paralytica, or syphilis, right? Using malaria. So these fevers that I'm telling you about all of a sudden had a very advantageous utility. You could give somebody malaria, they would have these fevers, the fevers kill the syphilis, and then you treat the patient with the drug. So everything seemed to work out fine. I wouldn't recommend it, but it was used for a long time. And then finally, many of you will be familiar with the fact that DDT made a big splash. It was extremely effective. The main reason we don't have malaria in the United States today is the use of DDT to eliminate these populations of mosquitoes that were transmitting the parasite within the United States. Okay, so we've talked a little bit about the disease. The disease is malaria, and the parasite that causes this disease is Plasmodium. And so Plasmodium are protists. It's a class of organism that is called an apicomplexa. What distinguishes apicomplexa, and there's about 5,000 different species of apicomplexa, is that it's a single cell. Okay, it's one cell. It's a single-celled organism. This organism has an apical prominence, so that puts it in this class apicomplexa. And this apical prominence attaches to different cell types. So there's 5,000 different species. The ones that we're particularly interested in that cause malaria all attach to blood. Okay, so this machinery here is involved in attaching to blood. All apicomplexa that have been studied today are actually parasites, and 5 of these infect humans. So all 5 of these different Plasmodium species infect humans. Plasmodium falciparum is the most virulent, lethal, deadly to us. That's the one that my lab focuses on the largest number of cases worldwide every year are due to Plasmodium falciparum. The one major take-home point. Newspapers butcher this all the time. It drives me crazy, so I'm going to just let everyone know. It's not a virus, okay? This cell is a eukaryotic cell, meaning that it is a highly complex cell. Its cells resemble our cells. It is not a simple little bacterium. It is not a much simpler virus. It's actually a very sophisticated cell. So the idea that I'd like to propose is that we need to understand how the cell actually functions, what makes it different than our cells. And those are the things that at the end of the day we want to actually be thinking about targeting. Okay, so it's a very complex cell, so we can't just think about it as this trivial little package that contains some information that we can manipulate. It's a complex cell, but that complex cell relies on our cells. It cannot live on its own. It always requires a host, and that's what makes it a parasite. There are numerous other related apicomplexa, some of which you might have heard about. So things that we learn about in studying Plasmodium and malaria actually translate to lots of agricultural issues. Okay, and many of these are actually of concern in the U.S. So there are apicomplexans such as babesia, which are transmitted by ticks. Malaria is a major issue for cattle, the cattle industry. Malaria infects chickens. Toxoplasma is carried by cats. Cryptosporidium is in the water supply and can infect a number of different hosts. So the general principles that we're learning about actually translate across, and there are people, of course, working on these other organisms, and we also learn a lot from the kind of things that they're learning in those organisms. Okay. One of the most fascinating things is, to me at least, is in fact that because this organism, this Plasmodium parasite, has been around so long, it basically infects every animal species on Earth. Okay? Birds, reptiles, mammals, you name it, there is some parasite that resembles this parasite that can infect it. Now, the reason I'm pointing that out is because A, that tells us something about how long this thing's been around, and B, it tells you that even if we wipe it out in humans, you better watch out, because if it's able to jump from one of these other species into humans again, we're going to have the same problem, and we won't be able to really identify it as the exact same thing. Okay, so this is the additional reservoir issue that has to be in the back of our minds when we think about what we're going to do. Okay, so I mentioned that the life cycle of this organism is extremely complex, and it's complex for the following reason. This is the full life cycle. What I'm going to focus a lot on today is this 48-hour blood stage. What happens here is that a single parasite infects a red blood cell. The red blood cell then allows for the parasite to mature. It eventually divides within one red blood cell and produces up to 24 new infectious particles. So in every round, you go from 1 to 24. That's a massive exponential growth of parasite. Okay, so you can imagine that in very few days, you're all of a sudden containing millions and millions of parasites. Okay, this parasite has to be transmitted to mosquitoes to complete the cycle. An obligate aspect of the cycle is a development first in the midgut of the mosquito. The parasite then traverses the midgut, develops on the outside of the midgut. They then become motile, so they have not only a free living stage, but they also have a stage that actually swims. It then swims its way up into the salivary gland of the mosquito. When the mosquito re-injects somebody, those forms are also motile. They swim through your vasculature and find their way into the liver. This entire initial process of having an injection and having these motile forms find their way to the liver is completely blind to your immune system. You have no idea that anything has happened. Yes, you've been bit by a mosquito, but no, you're not responding to the parasite. It then, essentially, of your entire liver, which is made up of millions of cells, it only invades one liver cell. So at that level, you're also not going to detect it. And from one infectious particle, it makes up to about 40,000 newly infectious particles. So you're totally blind to what's going on at this point. And all of a sudden, they break out. You have 40,000 of these parasites that come into your bloodstream and start invading the blood and doing this cycle. Okay, so the numbers are staggeringly high, and it happens very quickly. So this liver stage takes about a week. This stage here, the mosquito, takes about 10 days. And this part of it is cyclical. And every 48 hours, there's bursting, reuptake in the blood, bursting, reuptake. So your body can't keep up with replenishing the blood that's actually being destroyed by the parasite. So now let's look a little bit more in detail. If we focus in on this blood stage, it's a 48-hour cycle, as I just mentioned. There are some important steps that happen along the way. First, you have the invasion of the parasite. So the parasite actually recognizes a red blood cell. It has to orient itself to work its way into the blood cell. It works its way in. It then establishes itself metabolically, turning on some early programs that have to do with eating some of the contents of the interior of the cell and bringing in nutrients from the outside. Then goes through a massive growth stage. So trophos means feeding. So it's feeding and growing. So the parasite's getting larger. Ultimately, it has to go through a division step. Schizogony means division. So it has a division step, and I mentioned already that one parasite produces up to 24 new parasites. So these guys then, once they're fully matured, have to actually break out, and you get this, you get these parasites bursting out and reinvading your blood cells. They also produce this sexual stage that I'm not going to talk about. It's really too much detail, but they have an alternative stage, and this is the only stage that can be transmitted to mosquitoes. So these other stages that you see here are really only part of the clinical disease and do not complete the cycle with respect to the mosquito itself. So I have a little video here of what this actually looks like. So what you have here, this is an infected red blood cell. For those of you who know anything about blood, blood's a fairly simple cell. It has mostly a lot of hemoglobin inside it to carry oxygen. Hemoglobin has a heme that is actually where the oxygen actually is carried in this molecule of hemoglobin. Parasites degrade hemoglobin and have to do something with heme, because heme is actually toxic. So they make this beautiful crystal. So you can see it actually with your naked eyes. It's this gorgeous little crystal that they make, and that's how they actually manage to survive. So what you're going to see here, this is a red blood cell full of parasites, and they're now going to work their way out of the cell, hopefully, if this will work. And if not, then we won't worry about it. That's too bad. Okay, so imagine a bunch of parasites coming out of this and then reinvading into the cell. And you see it's all about the close proximity. So you always have very high quantities of red blood cells around. These guys can easily reinvade. And in fact, they have to do so within minutes or they get cleared by the spleen. Now what's interesting is this process takes you from a normal-looking red blood cell very quickly to these highly deformed cells. Okay, so these deformed cells, one of the special properties of the deformation that occurs once they're invaded by parasites is that, in fact, they become sticky. And the reason that Plasmodium is so virulent or so deadly is, in fact, that this stickiness is their key to success. They no longer circulate once they've been invaded by a parasite. The parasite makes proteins that it puts on the surface of the red blood cell, and that red blood cell sticks to your vasculature, so in your capillaries, it just sticks and no longer freely moves throughout your body. So the spleen can never detect it. Okay, that's how it evades the immune system during its 48-hour cycle before it gets released again. So it has this very sophisticated means of actually doing this adherence. The way it does this is by putting these proteins out there, and what that actually does, if you look in very high resolution at the surface of the red blood cell now, it has these little knob-like structures, and each one of these is loaded with a bunch of sticky proteins that ultimately form this adherent surface and take it out of circulation. So most of you have probably heard of sickle cell anemia. So sickle cell is another one of these very interesting aspects of our sort of genetic interaction with the malaria parasite. It tells us also that we must have been interacting with this parasite for a long time, because our genetics altered itself so that we could actually deal with this. If you actually have one good copy and one bad copy of sickle hemoglobin, you live your life totally normally. And in fact, the advantage is that you can be infected by malaria parasites and not ever get to high enough titers to kill you. You're the perfect carrier. You will always carry malaria parasites, but it does not get to the point where it can actually kill you. Okay? So that is a perfect human adaptation. If you end up with two bad copies of this, you're in bad trouble, because now you have sickle cell. And sickle cell is a devastating disease. And so, of course, in these areas, you know, with some frequency, you end up with a much higher frequency of people with sickle. Okay, as I mentioned, there's further development ultimately in the midgut of the mosquito. And this just shows that the process really is initially in the midgut about a 12-hour process of development. These parasites undergo motile and non-motile forms, which is, to me, very fascinating how they can continue to change their machinery. Once they actually end up on the outside, so they swim their way across these cells, they develop out here for another 12 to 20 days. And again, they become motile, they swim up to the salivary gland, and the mosquito can then inject them. I assume that this video will also not play because I don't know what's going on. But this is a really cool video. I have video at the end that actually works. This video here, what you're looking at right there, this is a remarkable video. I love showing this because it's that is the tip of the mosquito proboscis. Okay, so it is actually injecting, in this case, a poor mouse's ear that has been stretched over a microscope, and you're watching this mosquito come in and actually inject the ear of a mouse. Okay, and what you actually see, you can capture everything really without the motion, but what happens here is that the mosquito injects these little particles that you see over here. These are the parasites, and these are these motile forms. So you can get rough estimates by taking hundreds of these videos of how many parasites are actually being injected, and it's a remarkably low number. So it's it's somewhere between 20 to 100 in any injection, and only one of these guys at best is ever going to survive. So the numbers game is remarkably low. It's totally against the parasite, but its main reason for survival is that at some point it explodes in numbers. So the infectivity rate is actually low, but the number of people being infected every day is very high, and certainly sufficient to keep this thing going in all of the endemic regions. Okay, so hopefully I painted a bad enough picture of what the situation is. Now we can think a little bit about strategies that have actually been used and are being used to combat the parasite. So there's three that I'll talk about briefly, just to give you a flavor of what's going on in the field. One is obviously drugs that target various aspects of the parasite and its development. The second realm where there's actually a lot of activity, despite the sort of bleak picture that I painted for you, there's a lot going on in development here. And the third is mosquito control, which I will continue to argue is certainly today the front runner with regard to actually having the highest success rate anywhere in the world. Okay, so if you can control mosquitoes, you're doing a great job. Now, these different types of control affect different parts of the cycle. So I've just depicted it slightly differently here. So the blood stage is mostly targetable through drugs because it has such a high number. So to target something with a vaccine when there's such a high number is going to be really hard. So drugs have been considered in this blood stage. Then in the mosquito, we have various aspects of vector control that I'll discuss. And ultimately here, when you're infecting from the mosquito to the liver, the hepatocyte is liver cells, that's where a lot of work is really going on looking at vaccines, because that's a place where there are a very small number of parasites. Remember, the mosquito is not injecting that many parasites. So if you could get something to recognize those guys and eliminate them, you would block the development. So there are a large number, there's a laundry list actually of different kinds of anti-malarial drugs that are either in use or have been used. Many of them are actually highly related. Okay, so quinine is one of the oldest drugs ever against malaria, and many of these highlighted here in blue are actually just derivatives of quinine, so slight modifications that are actually also active. Then we have others that target other different pathways. It's not really important. And the bottom line is that currently today, the most effective way to treat people is to not use any single dose of anything. You always combine drugs. And that's sort of the classic way to get around the issue of resistance. So I'd just like to highlight quinine, in fact, has been around for a long time. It was first reported in 1683 and has been used extremely effectively, has had tremendous impact in many wars for people interested in world history. Whoever had good supplies of quinine was going to win. Okay? Napoleon has had tremendous success, just pitting armies into malarious regions, never attacking them, just surrounding them, and just waiting for them to die on their own. Okay, you never have to fire a single shot. You just wait long enough, the mosquito will do all the work for you. All you need to do is know where the malaria is and have quinine. And so quinine, it comes from the bark of this tree, was grown all over the world at one point. And the problem with that is ultimately it led to resistance. Now, of course, quinine is disgusting, And so that leads to the formulation here, the classic gin and tonic, as a great way to get your quinine. In fact, you buy tonic water today, it still has quinine in it. Okay, that gives it this nice little taste that people like. All right, it's a slightly bitter flavor that is quite appealing to some people, and more appealing with gin. Okay, so the bottom line is that what you're looking at here, of course, is a world map. In red are all the regions of the world that are predominantly afflicted with malaria today. It's sub-Saharan Africa, Central America, South America, and Southeast Asia. All the places that have dots on them are places where there is known and established resistance to, at that point, the front runner drugs. But you can have a similar map for every drug, essentially, that is or has ever been used, okay? And the bottom line is, despite our best efforts and massive worldwide campaigns, malaria is on the rise, okay? It is not going down anywhere. Okay. So, back to my pet peeve. Malaria vaccines, all right? I get emails from people all the time telling me they just read about the most phenomenal malaria vaccine, and you know, it's great when people out of the field know more than those of us who are in the field, but the media seems to be very quick. The bottom line is there is no current vaccine, and the main reason really is that most vaccines are actually based on cells that were once alive. Okay, so you need a living cell. Normally, you take a living cell and you make it not quite grow as well as the thing that's going to infect you, so your body can develop an immune response to it, so that when you see the real thing, it knows to attack it. Okay, that's how most vaccines are established in an overly simplified manner. So that's very hard to do with Plasmodium, and the reason is the following. The main place that I mentioned that you want to look for an actual candidate for vaccine is in the mosquito, okay? Here's my little mosquito, okay? Really hard to see under the, like aspects of the mosquito are really hard to see under the microscope. Here's a little bit of a blow up. Here's where the parasite actually resides. This is now somebody's tedious work of taking a mosquito, putting it under the microscope, dissecting it apart, pulling out the salivary glands, and then if you carefully squeeze the salivary glands, you can get out the parasite. They're called sporozoites at this stage. And actually, that would be what you want to inject into people. Sounds pretty easy, right? So all you have to do is get a bunch of that material, because you can't make it any other way. So somebody actually made a company that's doing this today. It sounds totally absurd, but there it is, Scenario.com. Look these guys up. These poor fellows, every day, they go into this room, and their job, so be thankful for whatever your job is or was or will be, they dissect mosquito heads and squeeze out the parasites from the salivary glands. And believe it or not, this is now past the FDA. Okay? You can actually inject this stuff into people ethically. The problem, obviously, that I'm sure everybody in this room is recognizing is how are you ever going to scale this up to the point where you're going to give it to either 500 million, 1 billion, 2 billion, many, many people. And I doubt that one dose is going to be enough. So that's their big problem at the moment. And the fact that when they did their first trial, despite the fact that they got a lot of press in the New York Times and everywhere else, it completely failed. Nobody was protected. Okay? So the media does a great job of hyping these things up. And the reason is, in fact, we know that this statement up here is true. If you take these guys and you actually X-ray irradiate them, you can inject them into people, and people can get protection. That experiment was done in 1972, and it worked. Totally unethical, but at that point in time, you could actually take prisoners, you could inject whatever you wanted in them, and this experiment was done, and it worked very well. The problem is that it's very hard to get that to be done in any sort of standardized format. Okay, so the first large-scale experiment from this company has totally failed. But there's hope. Okay, there are millions, millions, millions of dollars being poured into this issue, and I actually think it's probably the best thing we have going for us with regard to a vaccine. So keep your eyes focused on that. The other, so there are other cool ideas out there. Here's one, and that is, here's one example. So why not focus on the mosquito? Okay, there have recently been a large number of papers where people are finding really interesting fungi that specifically kill the mosquito that carries the malaria parasite. Now, I don't know how they're going to deploy this, but you can actually do this very effectively. It is specific for this mosquito. It infects it in such a way, as you can see here, that in this is a number of, I think it's 2 or 3 days, mosquitoes are dead. They need to survive at least 14 days for the malaria parasite to go through its whole cycle. So maybe something like this is in the future. I have no idea. Bottom line is that the most effective treatment today is still bed nets. Okay, we have treatments. You and I, if we travel, we can all afford treatments. There are good treatments. They're expensive. And the benchmark, believe it or not, is chloroquine, a derivative of quinine. When that was the best drug in the world, it cost a penny a dose to make it. Okay, you can't even make a penny for a penny. And you have to now make a drug for a penny. And that's where it has to, that's the target for those of us in this field. Is we need to come up with an idea that can be produced at that cost. And the numbers are billions and billions of dollars because the number of people who need to receive these medications are in that realm. Okay, so that's sort of the challenge. And the simple solutions are these. Has anyone ever slept under a bed net? How comfortable is that?
It's fine.
It's fine depending on where you are, yes. If it's 100 degrees outside and you're under a bed net, that thing does not breathe at all. So they make great fishing nets. They're great in windows. They get used for all sorts of things. And in fact, places where they're used, they're phenomenal. Malaria rates have dropped to 0. It's remarkable. It's absolutely remarkable. The parasite no longer has, the host. If you eliminate anybody carrying this thing, it's eventually going to disappear from any community. It's a great solution, but it's not foolproof. You cannot ever implement it widely enough, I think, to actually get it to have the effect that is desired. Okay, so that's my background. Here's my motivation. All right, and this is getting worse every day. So since I started my lab here at Princeton, about 6 years ago now, over 7 million children have died. Okay, I don't like this idea, but very soon we're going to be at the population of New Jersey, which is on the bottom, right? And that number is just going to go up and up and up, and it's been going on forever. Most of us never think about this, one of the things that I always struggle with is, why aren't these numbers alarming to people? And yet when it happens in one shocking event, there's worldwide outcry and outpouring of money and to deal with situations that are very acute, and yet problems that sort of plague us constantly every day are highly ignored in the big picture. Okay, so enough of my soapbox. What are some of the big issues that are being thought about in this field? And now I'm going to try to get into some of the things that we're actually doing in my lab and try to relay some of the ideas that are being thought about in the field in general. So the first one is, the parasite, as I mentioned, requires the host. Okay, it requires, in us, it requires blood cells and it requires liver cells. Those are two fundamental stages that it requires. It then also requires development in the mosquito. And so we should understand better how that interaction actually occurs. How are those cells recognized once the parasite gets in? What is it doing in there? What is it providing and what is the host providing? Now, if you can figure out those parameters, maybe you can actually block the development of the parasite. The second is that an infected cell is somehow blind to the immune system. Okay, how does it do this? So I mentioned one way it does it is that it sticks and it doesn't circulate anymore. That's true to some degree. But eventually your immune system, there's millions of cells that are infected. It's going to start figuring this out. The parasite has this really sophisticated method of dealing with this, and that is that it changes which protein it puts on the surface, so that once you figure out what it is, it's going to put another one out there. And it's just as sticky as the previous one. And so it's constantly doing this thing that's called antigenic variation, which leads to this immunization. I mean, this is a big problem, but if we can figure out how it does this, how is it doing this switching? What if we made it put all those proteins on the outside? Then that should be a pretty good vaccine maybe, because your immune system would recognize everything that's ever on the surface. But we haven't figured out how to do that. Okay. So in red are the things that I'm working on. First is the regulation of these developmental cycles. What I mean by that is what actually tells the parasite how to progress from one stage to another when it's within the blood, and we'll get more into that. And the second is to think about metabolic pathways. So parasites, like any cell, need to take one molecule and convert it to another molecule. These either become building blocks for the cell, or they're food for the cell, or they convert it to energy, just so that cells can grow and ultimately divide. That's the function of all cells, okay? So we don't know what those pathways actually are, what the process of making these molecules is. And this is where I'll get a little bit into this metabolomics idea, which is to try to capture what is actually going on within a parasite. Okay, the other thing is that we'd like to understand certain specific aspects of parasites, and I put here this thing called a plastid. What is that? That is an interesting little organelle that's in the parasite that is actually derived from a plant. So you and I never would have this plastid thing because we have no direct link to what's going on in plants. But because at some point these parasites in their evolutionary lineage acquired a cell that has a plant-like lineage, they have very interesting biology that's associated to plants. The reason this is interesting is that if you can target something that's non-mammalian or non-human, that will always differentiate in a sort of pharmaceutical manner or by drugs. It'll never cross-react with what's going on in your cells. And that's the ideal scenario. You want drugs that hit only parasites, don't hit your cells. Okay, most drugs on the market, it's this game of sometimes they're affecting your cells, but more of the time they're affecting the other cells that are in your body, so they get eliminated before your cells get eliminated. And that's a game that we're willing to play because at the end of the day we feel better. And that's okay, okay? But in this case we want to try to differentiate as much as possible to avoid resistance as well. And I'll skip the last point because it's not that important at the moment. Okay, so why are we able to actually think potentially more deeply about this situation today? And there's a few things that I think are really important. The first is that we actually now have a better understanding of the parasite at the genome level. We know what all the genes are, all the proteins are within this cell. Okay, so we know all the building blocks of the actual cell. And that's really important because now we actually have a catalog of all the possible things that we think the cell might do. The only problem with that is, in fact, that 60%, I don't know if you guys can read this, but 60% of the genes, or the proteins that it has, or these building blocks, are unknown. We've never seen them before. Okay? Most people think that's a problem. I think that's an opportunity. Let's figure out what the heck those things are. Okay? If we figure out what those are and what they're doing, those are probably the things that we actually want to be thinking about targeting. Okay, so that's what I spend all my time doing, is figuring out what these genes of unknown function actually do in malaria parasites. Okay, so let's talk about metabolism for a minute. This is meant to confuse you. It's just a bunch of random lines, right? In fact, this is a network diagram of the kind of chemical reactions that are actually occurring in your cell. This network diagram is actually very similar regardless of what kind of cell we're talking about. Okay? Most cells resemble each other in very basic processes. At the top, in blue, is something very fundamental to all living cells. They need some way to take in carbon. We take in carbon in the form of sugars, mostly. We digest sugar, so each one of these little nodes along the way is actually a different reaction that's catalyzed by an enzyme that's converting a sugar into some byproduct of the sugar, in some cases releasing energy, in other cases making other molecules that you might need for new cells. And all that this does is show you that, the main thing is you need to come down this path. You spin off all kinds of things off this path. At the end of the day, this circle down here is actually pretty important. I'm going to talk a little bit more about this. This is where you make all your energy. But all these spin-off reactions are utilized to make all the basic building blocks that a cell needs. So amino acids make proteins. Nucleotides is so you can copy your DNA. Lipids is so you can make new membranes for your actual new cells. Cells have to turn over. You need new cells as you grow. And so all of this is just sort of fundamental to any given cell. Now, our understanding of these processes is pretty good currently. A lot of this leads to many of the common pharmaceuticals that we have today. And sort of at the basic level, as I said, a lot of this is complementary from cell to cell, so we can translate that information back and forth if the enzymes or the nodes are similar in different organisms. That's true to a particular point. Now, the other reason this is so important is that most drugs currently on the market today target metabolism. Okay? It's based on our fundamental understanding of metabolism, and drugs target, for example, in this case, an enzyme that's involved in using this particular molecule. If you just change a single atom over here, so I added a fluorine out here, this thing now is a very potent anti-cancer drug. Okay, so it's a very minor change chemically, but it has a massive effect on cancer cells. Okay, it basically disrupts the production of functional DNA in that cell, and these cells ultimately die off. Okay, it looks very trivial, right? But if we didn't understand the process, we would never have arrived at this molecule in the first place. Okay, so the idea is that many of the targets of drugs are actually enzymes, or these nodes. So how do we figure out what's going on within cells? So this is another buzzword term, systems biology. We have to use it because it helps us get funded. But in fact, systems biology means different things to different people. And to me, it basically just means the integration of all data that you actually have available to you. So I'm not going to belabor what these different aspects are, but there's a lot of work in this sort of central dogma of biology, which is moving from DNA to RNA to proteins that then do what the cell actually has to do. But proteins all work on small molecules, and small molecules are generically classified as metabolites. So what we want to do is measure the metabolites, or the small molecules, in any given cell. Okay? The reason we haven't been able to do this is because the technology hasn't been there, but now it's there. Now, if I didn't have the technology, what I would do is I would draw a generic. This is now the wiring diagram, the same thing I showed you before that was much more complicated, but with a few more bells and whistles, that's now Plasmodium specific. And this was drawn up after we knew what all the building blocks of the actual parasite were. Most of these lines are actually wrong. Okay, they're just inferred because the parasite has the building block to maybe do what we think it does. So the lines are arbitrary. We're just connecting one molecule going to the next molecule, and this is that same pathway that I showed you before, where you take in sugar or glucose, you convert it into something else, you turn this cycle down here, you make energy. All this is wrong. Okay? Why is it wrong? It's because we're inferring biology from what we know in a bacterium and what we know in a human cell, and assuming that it's the same in parasites, and that's bad business. So that's cool. That gives us a whole new realm to work in. And the reason that we can do this is because of the technology. So the technology is this fancy machine here. It's a mass spectrometer. I don't need to go through the details, but the important feature that I want to highlight is that what we can do today is we can take parasites and we can actually break them open, release all the thousands of different molecule types that they have, and analyze each individual one of these molecules. And I can figure out how much of each one is in any given cell at any point in time. Okay, now that's the main feature of what I need to get across with this. This machine has extremely good accuracy, and it can tell molecules apart with very good accuracy. And when I use this type of approach, and I go back to this map, everything highlighted in blue here can basically be measured in 1 hour using this machine. So it takes me 15 minutes to make the extract, and now I can run it on this machine, and I can figure out what all these metabolites are. Now, what I'm showing you here are the ones that I presume should be there. But there are hundreds to thousands of other molecules that are also in there. That, I think, is the interesting and the important part. Not what we think should be in there, but what we didn't know was in there. And if those molecules are in there, they have to be made by the parasite. Okay? So let's see if you can follow this a little. What this allows us to ultimately do, if you can measure what's going on in a cell, you can start asking these basic questions that I talked about. How does the host being the blood cell, perhaps, interact with the parasite? So the cell's full of molecules. Which ones does the parasite want to take up and use? And what molecules does the parasite make that it spits out to the red blood cell that the red blood cell uses? This is an exchange. This is why it's a parasitic relationship that allows the parasite to actually exist and allows it to go undetected by the immune system. Secondarily, we can actually use this to now look at particular pathways, and I'll give you one simple example of how we do that. Third, we can look at the actual metabolic response to known drugs or anti-malarial compounds. You are probably not surprised that for most anti-malarials that people take today, we have no idea how they work. That medicine doesn't care, in fact, how drugs work. You don't have to know how a drug works. You just need to know that it does better for you than it does at killing you. That's it. There's a very fine line there. And most antimalarials, we don't have any idea how they work. And I'd like to argue that it'd be informative to understand how they work, because then we can make them better, or at least know where they're actually reacting so that we can target 2 different places at the same time and avoid cross-reactivity, avoid the emergence of resistance. Okay, I'll skip this point and I'll get to this last point. Identifying unknown compounds. Identifying unknown compounds, I think, is where the ultimate end game is here. If I can find molecules that are being made, then parasites have to make it. The way any organism makes a molecule is that it takes another molecule and it changes it to that molecule. Okay, that's chemistry. The chemistry is very slow, and so all cells use enzymes to do their chemistry and to accelerate it. All right, so if I can detect the molecule, that means that there's some protein that made that molecule, and that means that if I figure out what that new enzyme is, I should be able to target it and drug it, especially if it's making something I've never seen before. So that's really the ultimate goal. All right, so here's a very simple example. Does anyone know what the tricarboxylic acid cycle is in general?
No.
No. Excellent. Okay, so back to this, if we go back to this diagram here, I mentioned that we eat sugars, we go through a bunch of reaction steps, and we normally make, out of a 6-carbon molecule, we make a 2-carbon molecule, and that gets turned through a bunch of reactions in this cycle that's always depicted as a circle, and this produces most of the energy that all of us use all the time when we do anything. Okay, this is they call the tricarboxylic acid cycle, or the Krebs cycle. It's one of the classic hallmarks of all living cells. Now, you can look it up in any textbook, and it'll look just like this. You come in with this 2-carbon molecule, and you basically add it and move it around to make these other molecules that eventually make energy. So the function of this cycle, make energy, provide substrates for other reactions, including including amino acids and additional biosynthetic precursors. And where you might have heard this happening is in this organelle here called the mitochondrion. So if you remember way back, whenever that might have been, the mitochondrion is the powerhouse of the cell. It's the powerhouse because it makes all the energy, okay? All the ATP that you need to do any of your daily activities. All right, so these guys have a mitochondrion as well. They also have all of the enzymes that are supposed to make this pathway actually work. And in fact, what we did is we took molecules and we labeled them. So now I can go and buy sugar that has a slightly different weight, because I now buy a sugar with 13 C glucose as opposed to 12 C glucose, and it has a slightly heavier weight. And I follow where do these molecules actually go when they're being utilized by parasites. And the bottom line is that they don't do this at all. Even though they have all these enzymes, they don't do this cycle. And so they basically don't produce energy this way. And in fact, what they do, I know this is really mind-boggling, right? Like, who cares? Right? Exactly, who cares? It's exciting to us because it's not a cycle. It's this new architecture for actually using these molecules. And what that means, at the end of the day, is that these enzymes function differently than has been seen in 99.99% of all other organisms ever studied to date. That's a pretty big deal. Nobel Prize was given for the Krebs cycle. Okay, it's a fundamental aspect of biology, and it's different in these parasites. That's cool. I'm thinking about how to target this particular pathway. Okay, so the bottom line is that I'm looking for unknown enzymes or enzymes that function differently to identify new, idealized drug targets. All right, that's metabolomics for you. Metabolomics 101, at least, for today. So, let's get back to the second part of what my lab works on, and that is a different aspect of fundamental biology, and that gets back again to the central dogma. How many people have heard of the central dogma before? Okay, a few. So the central dogma of all cells is that your cells encode DNA, right? DNA is the genetic code that carries the information from cell to cell, from progeny to progeny to our children, etc. And the reason that that's important is because it encodes all of, ultimately, the proteins that are down here. And the processes that do this is you first take DNA, you convert it to mRNA, a messenger RNA, that then gets made through translation into a protein, and proteins do everything that our cells need to do. Okay, so this top process, this conversion of DNA to RNA, is an extremely well understood process in all organisms. It's fundamentally similar in all organisms. And the way it works is that some protein binds to some place in the DNA and tells it to make a copy of this particular gene that will ultimately be your protein that's functional. And the way this is done is through these blue DNA binding proteins. Now, my lab, for the first time, has identified the first and unique class of DNA binding proteins that have a particular name that doesn't matter, but they actually turn out to be very interesting because they're not only found in Plasmodium, but they're found in all other apicomplexes. So all these agriculturally related parasites, they all contain these DNA binding proteins. And what's exciting about that, it's really the only set of regulators that are present at all to tell cells how and when to go through these programs. Okay, now, so I'm focusing very much on this 48-hour cycle to understand what regulates this progression that you see here. Forget about the details over here, that's not important. But what we've done is we've characterized this actual, this protein. And we can see on the top in blue and red are two different molecules of the actual protein. In green is the DNA. And you see how it sits on this DNA as I cartooned before with that blue circle. And this then tells a whole lot of other machinery to come to this spot and make a particular gene so that you can make a protein. Now, why do I care? Because this protein is not found anywhere in mammals. It's not found in humans. it has a plant origin. It's completely different than anything that's ever been seen in humans. So the idea is, can we block its interaction in this fundamental process? So no organism can survive without this kind of regulation happening. These cells are doing it differently than we do it. Can we actually co-opt this and block it? Now, I told you that my lab, I'm not going to go through the details, but we came up with this structure of what this interaction actually looks like. And, you know, I had the idea that, you know, blocking this might be interesting. I never really thought we would be able to make much headway. And in fact, a group then from Denmark calls me up one day and is like, you know, we've been running some computer simulations on that structure that I just showed you. And we have hundreds of thousands of molecules that we can then take and in the computer basically make it interact with your protein, find the optimal interaction, and then see if that interaction is more favorable than the interaction of my protein with DNA. And so at the end of the day, they're trying to see if the interaction of some small molecule is more favored with the protein or more favorable for DNA. And they come up with a short list of 12 molecules, 11 compounds, sorry, that seem to interact better with this than my molecule with DNA. I'm like, yeah, so what? I can come up with a script that can do the same thing. That, you know, if I just tweak the parameters just right, it's also not going to interact. It's purely in silico. You know, what does that actually mean? So they send me all these pretty pictures. They're showing me where it might bind. There's no data here. Okay, this is just what the computer thinks is probably the best fit. So I got more interested when they told me, you know, most of these compounds are actually known therapeutic agents. So if any of them actually work, we can just pull them off the shelf, bypass years of work that would need to get us to the FDA, and actually see if we can get something to market a little quicker. And the way we tested so they eventually convinced us to do something about this. So the way we do this is very simple. Let's see if I can actually convey this one experiment. So what we do is we have a piece of DNA that has a certain sequence. Okay, these are the DNA bases that are recognized by the protein. If a protein binds to this, it will move it from this location up to this location. All right? So this is what normally should happen, is that protein interacting with DNA should move from here to here. If we add the molecule, in this case it's called Prussian blue, in increasing amounts, what you're seeing is this band is going down as we increase the amount of drug. And lo and behold, at some amount of drug, there's no interaction anymore. I couldn't believe it. It's like, go and do it again. It came back and it worked over and over and over again. In fact, it worked on almost all of the compounds. And then, the important thing is, does it actually kill parasites? So we set up the little in vitro system, and it kills parasites. There's a broad range. This is the value at which they're killing parasites. It's not incredible. It's nothing that's going out to market anytime soon, but it's a great starting point. And now we can interact with chemists to make these molecules better, to see if we can actually disrupt this and make this a more efficacious drug to kill parasites. So this brings me to near the close of my talk. I wanted to stress today, I wanted to get across something about the malaria parasite and its general life cycle. But what I really want to emphasize, and what you should realize, is that the majority of research today really is a spin-off of other research that's been going on for a really long time. And I think that's a bad way to do science. So there's a whole realm of biology that's untouched. And it's untouched because there's no good presumed starting point. Okay, so the literature basically focuses on 10% of the proteins that we know about in the vast array of all enzymes that are out there. That means there's a huge amount of space which we haven't looked into and which we're not taking advantage of with regard to new approaches. It's riskier, it's harder, you don't know where you're going, you don't have anything to base it on really, but I think that's where all the exciting stuff is. So that's been my approach to doing this. And I just want to close also by saying that while I started this out saying that there's not really a whole lot of attention on malaria today worldwide, it's not exactly true. In fact, the fact that organizations like the Bill and Melinda Gates Foundation have stepped in have made a tremendous impact on this field. For the first time, there's serious private money. I get no money from them, by the way, so I'm not making a pitch for them. But they have put it back on the map. And what that has done is it's gotten our government interested again. And they don't want to just see the private sector getting involved. They want to have a direct impact in this sort of work. And a lot of these other initiatives are actually directly funded by the government through the NIH or the White House or other different aspects. And so it's a pretty exciting time. Never has there been so much research actually ongoing in this particular field. And so it's a really exciting time to be involved. And with that, I just want to make sure I thank the people. This is the lab. These guys do all the work. I just do this stuff. And these are the funders. I thank you for your attention, and I'm more than happy to take any questions you might have.
Humans have guts. Malaria, mosquitoes have guts. There has to be something different between the kind of gut that a human has and what a mosquito has that requires that you go through the cycle in the mosquito.
Right.
Is there any understanding of what the differences might be?
Yeah, in fact, so one of the main differences is that I showed this very specialized form of the parasite that is what actually gets taken up by the mosquito to survive the mosquito. Those were these, anyway, I'll get back to it slowly. This is a form that undergoes a large amount of differentiation, specifically in the mosquito, because it's triggered by two things. It's triggered first by a drop in temperature, and that is obligate. It has to be, it's required for the parasite to actually undergo that transition. And secondarily, so it's this image here where this form here, this gametocyte, or this sexual stage, has to be taken up. So it is totally dead if it stays in our circulatory system. Okay, it can only transmit once it's in the mosquito. And that process of maturation requires a temperature change, and it requires a pH change that's only present once it gets in the midgut of the mosquito. So, and there's two molecules that will mean nothing to anyone, but these two molecules also induce this transformation event that has to occur there and would not occur in our midgut, for example. Yeah.
So when you were in Africa, when you're thinking Malaria, how does something like Malaria work to prevent you from becoming infected with?
Oh, it doesn't prevent you from becoming infected.
What does it do? Our daughter's there right now.
Yeah, sure. No, I mean, I've taken Malaria myself. No, I've taken Malaria myself. I mean, you can still get infected, but what it's going to do is it's going to block it very early on at this, at the once it releases from the liver stage. The numbers are actually manageable at this stage, and it's a very effective drug. So you never initiate this 48-hour cycle that is essential to really get into any sort of full-blown infection. So if you're taking it prophylactically, you're never going to actually develop infection. Yeah. So you don't need to worry. Malaria currently does not have a high amount of resistance because it's a combination therapy. Yeah, in the back.
Wouldn't it be important to control the mosquitoes? And
Absolutely.
Perhaps simpler?
Yeah, so a lot of work is actually going on in mosquito intervention. We all know DDT was very effective. There are massive campaigns against it. And in fact, those countries that were encouraged to stop using DDT are starting to use it again because of its efficacious nature. There is just nothing better than it yet. And other modes of controlling mosquitoes include releasing infertile mosquitoes that will breed would then produce infertile offspring. Those types of experiments have been done on a small scale that kind of work. Again, the question really is how sustainable is that? Can you actually do that large scale? Nobody's been willing to actually go there. But yeah, I think it's a very, very good point that looking at the mosquito is just as important as looking at the parasite itself. Nobody wants to be around mosquitoes anyway. Yes?
How many people around the world are involved in this kind of work? If you have critical mass, everybody can tell each other, you know what you're talking about.
How many,
Well, how big is the community?
There's easily 200 labs worldwide. Yeah, I just came back from probably the biggest meeting, and that was in Australia. The Australians are actually at the very top of this game. They're tremendous, and yeah, somewhere around 200. In the U.S., it was a much smaller number until recently. So there, probably 2 decades ago, there were 5 to 10 labs. Of course, in the U.S., it's a very well. It's the same anywhere. The main interest was always through the armed forces, because those are the people who are going to have to send people into malarious regions. Otherwise, it wouldn't really be a concern. But that's what drives a lot of the research internally. Sorry.
How feasible is it to eliminate malaria on a country-by-country basis, or is it just going to spread all over again?
Yeah, so again, that brings up a very good point. The challenge of having 2 countries next to each other, one being slightly better off economically and more able to divert funds into tackling this sort of issue, being next door to another country that can't do that, forget it. You're just never going to solve the problem. It's going to keep coming across. Well, mosquitoes fly, and wherever a mosquito can go, you can continue having a problem. And so it's not so much about the people. And in addition to that, it's one of the interesting aspects of malaria, I think, is that most people in endemic regions are not sick, but they are carriers of vast quantities of parasite. Okay, so the parasite, you can go to these areas, and if you do a blood smear and you look in their blood, they have parasites. They never present as being sick, but they're the ones that are the real problem. So at the end of the day, whatever solution we come up with, you're not just going to be treating sick people. You have to give it to everybody. So, it's just like a polio eradication campaign or something like this. Every individual around the world would essentially have to get doses of whatever it is that we come up with.
Why are they not sick?
Why are they not sick? Oh, because there's the immune system kicks in and keeps them at a certain level, and because of this ability of theirs to switch, they just, they never proliferate to high enough titers to actually make you sick, because the immune system keeps them suppressed. And I think that's part of the real ingenuity of the parasite, is that it can keep dividing. It doesn't need to have large numbers, and it knows essentially when to influence those numbers. It's very well documented, right? One of the main reasons that it has to do this, irrespective of the fact that it has this interaction with the human host, is the following. So many of these regions have wet and dry seasons, okay? If the dry season was sufficient to eliminate the parasite, we probably would have done much better at this a long time ago. During the dry season, everybody's still carrying mosquitoes, okay? I'm sorry, everybody's carrying parasites, okay? As soon as the wet season comes again, mosquitoes come out, and all of a sudden, people's parasite titers go up. So some signal from mosquitoes is now telling parasites in all these carriers, make more parasites so that we can transmit more parasite. So it just happens over and over again, right? So you have to get through these long spells of the dry season. It just lays low. It doesn't lay low in mosquitoes, it lays low in humans. And so most people in endemic regions are infected and do not have any symptoms whatsoever. Therefore, the deaths are occurring in small children and essentially in pregnancy, because it's just the amount of blood that's associated with pregnancy and the placenta, et cetera, is, you know, it overwhelms the system. So those are the two main clinical manifestations. Yeah.
Based on what you were just saying, you know, carrying it, with so much international travel, when you think enough people would travel that it actually would spread back from South America?
Yeah, we don't have the mosquitoes
Do you have
anymore.
those specific mosquitoes? I mean, we've done this.
Yeah, we just don't have, so the mosquito has to be it's very particular.
It has to be.
It has to be that mosquito. It has to be the Anopheles mosquito. There are several species of it, but it has to be those mosquitoes.
And the mosquitoes don't fly as far.
No, they'll appear every now and then. They do exist in the U.S. There are cases of malaria every now and then in Florida. And Jersey used to be a big malaria area, right? We have the swamplands. Like, there's lots of mosquitoes here in the summer. We had a major problem with malaria. It's just, again, you eliminate the source for long enough, and it does go out of the system. I don't know what the numbers actually are in Jersey, but when I was working in California, I know that the numbers in California that are spent in just mosquito control is $1 per person. That's a ton of money. Okay, that's actual, like, every year on their budget is $1 per person to just deal with mosquitoes. None of the countries that we're talking about could ever dream of having that kind of campaign going on daily. And that's not even DDT, that's just like, you know, putting different fish in certain ponds and doing other tactics that actually get at the situation to keep the numbers down. It's just unsustainable and with the current approaches that we have. Yeah.
Next question.
I was just curious about why Europeans tend to use Lariam less and seem to have a negative attitude towards Americans going to malaria-affected regions and taking Lariam. Is there something about the interaction with the environment that makes Lariam less desirable? Besides,
Who has this problem?
Europeans. I mean, they tend to take the mixtures of drugs, and I didn't know if it was just effectiveness or there's some perception that Lariam has some causes more.
Yeah, so Lariam is one of these drugs that will have the potential for side effects. And that's why and Lariam is the most commonly prescribed drug in the U.S. For travelers. You're always told to take it before you leave. That's not to build up some amount, it's so that you figure out if you're going to freak out on it or not. So if you ever take this stuff, do take it before you go, because the last thing you want is to have these effects. It affects a very small percentage of people, but if you're one of those people, you want to know before you leave home that you want something else. I haven't heard this specifically. My guess, my hunch would just be there are different companies making different drugs. There are American companies, there are European companies. It's a market. I don't know what's driving that. I've never heard that.
Thank you very, very much.